Angiosperms. Unlike the gymnosperms, where the ovules are naked, in the angiosperms or flowering plants, the pollen grains and ovules are developed in specialized structures called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest Ulfia to the tall trees of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into two classes, the dicotyledons and the monocotyledons. Figure 3.5. The dicotyledons are characterized by seeds having two cotyledons, reticulate venations in the leaves, and tetramerous or pentamerous flowers, that is, having 4 or 5 members in each floral wall. The monocotyledons, on the other hand, are characterized by single cotyledonous seeds, parallel venation in leaves, and trimerous flowers having 3 members in each floral walls. The male sex organ in the flower is the stamen. Each stamen consists of slender filament with an anther at the tip. Within the anther, the pollen mother cell divided by meiosis to produce microspores, which matures into pollen grains. The female sex organ in a flower is the pistil. Pistil consists of a swollen ovary at its base, a long slender style, and stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cell that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and 1 divide to form the embryo sac. Each embryo sac has 3-celled egg apparatus, 1 egg cell and 2 synergids, 3 antipodal cells and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain after dispersal from the anther are carried by the wind or various other agencies to the stigma of the pistil. This is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tubes grow through the tissues of the stigma and style and reach the ovule. The pollen tubes enter the embryo sac where 2 male gametes are discharged. One of the male gamete fuses with the egg cell, the syngamy, to form a zygote. The other male gamete fuses with the diploid secondary nucleus to produce the triploid primary endosperm nucleus, that is PEN. Because of the occurrence of 2 fusions, that is syngamy and triple fusion, this event is termed as double fertilization, an event unique to angiosperms. The zygote develops into an embryo with 1 or 2 cotyledons, and the PEN develops into endosperm, which provides nourishment to the developing embryo. The synergids and antipodals degenerate after fertilization. During these events, the ovules develop into seeds and the ovaries develop into fruit. The life cycle of an angiosperm is shown in figure 3.6. Figure 3.5. Angiosperms. A dicotyledon. B, a monocotyledon. Figure 3.6. Life cycle of an angiosperm.